The human brain sits inside a completely dark, silent, sealed cranial vault. It has no direct physical contact with the outside world. Yet, to keep the body alive and maintain homeostasis, the central nervous system has to constantly monitor a highly volatile external environment. It must track incoming electromagnetic waves, which we naturally perceive as light, and mechanical compression waves, which we perceive as sound. This creates a strict physiological paradox. The brain cannot directly process a raw photon, nor can it compute an acoustic wave. Its only functional language is electrochemical impulses. Therefore, external physical energy must somehow be converted into this internal neural currency. In biology, we call this essential process transduction. Sensory organs function as highly specialized, multi-chambered transduction engines to bridge this gap. The human eye and ear represent the body's two most complex sensory processing pipelines. Look at this diagram detailing the internal structure of the eyeball. Notice its spherical architecture, measuring about 2.5 centimeters in diameter, wrapped in 3 distinct compartmentalized coats. Now look at the internal structure of the ear. Instead of a sphere, it is a deep 3-part cavern bored directly into the temporal bone of the skull. To understand how these organs solve the transduction problem, we will track a single photon and a single sound wave through 3 distinct functional phases: capture, amplification, and transduction. Despite processing different physical inputs, both organs rely on nested, heavy protective architecture on the outside to isolate their delicate transduction interfaces deep on the inside. Returning to the diagram of the eye, phase 1 begins at the outer fibrous coat, protecting the organ and capturing initial light rays. The transparent cornea provides the first stage of focusing. Light then passes through the pupil into the crystalline lens. The exact amount of light entering the chamber is regulated by the muscular iris. To ensure the optical path remains clear, the structural space is pressurized and maintained by transparent fluids, the aqueous and vitreous humors. Over in the ear, the capture mechanism begins with the auricle, or pinna. Its funnel shape collects mechanical sound waves from the air and directs them tightly down the external auditory meatus, right up to the tympanic membrane. In both organs, this first phase is strictly mechanical. The systems route and concentrate external physics without altering the signal's properties. Once inside, the physical signals have to be conditioned. In the eye, this occurs at the middle vascular coat, also known as the uvea. This animated cross-section shows the hazard of a fluid-filled chamber. Without conditioning, photons would continuously scatter and reflect off the inner walls, degrading the image. To counter this, a highly vascular layer called the choroid utilizes melanocytes. These cells produce a dark brown melanin pigment that absorbs excess light, reducing internal scattering and keeping the incoming projection sharp. The middle ear faces a different hurdle, the loss of physical energy as a sound wave moves from air into fluid. To bridge this gap, the tympanic membrane connects to 3 tiny auditory ossicles, the malleus, incus, and stapes. This bone chain acts as a highly efficient lever system. It translates broad, low-pressure vibrations hitting the eardrum into highly concentrated, high-pressure mechanical force. This amplified force is then delivered directly to the inner ear through a strict boundary point known as the fenestra ovalis, or oval window. The eye darkens its internal chamber to sharpen the signal, while the ear utilizes a bone lever system to forcefully amplify it. Both signals are now prepped for the final phase. We arrive at the third innermost coat of the eyeball, the inner nervous coat. This extremely thin, transparent membrane is the retina, housing the actual sensory photoreceptor cells, the rods and cones. Rods dictate dim light vision using a highly sensitive chemical called rhodopsin. Cones require bright light to activate, utilizing different pigments to parse out red, blue, or green wavelengths. When incoming light hits the retina, it physically bleaches these chemical pigments. That specific chemical reaction is the moment of phototransduction. The photon's energy triggers an electrical impulse. These new impulses route toward the optic disc, which creates our natural blind spot, and exit the eyeball entirely via the optic nerve. Back in the ear, the final stage happens deep inside the temporal bone, within a structure known as the labyrinth. This consists of a bony outer shell containing a membranous inner tube, all suspended in layers of perilymph and endolymph fluids. Look closely at this animation of the cochlea. When the stapes strikes the oval window, it creates physical waves in the fluid. These waves displace a central divider called the basilar membrane, forcing tiny hair-like projections in the organ of Corti to bend. That mechanical bending of the auditory receptors is the exact moment of mechanotransduction. The physical vibration is converted into a nervous impulse. These impulses leave the cochlea through the 8th cranial nerve, completing the journey from an atmospheric acoustic wave to interpretable brain data. The 8th cranial nerve actually carries 2 distinct streams of data. Hearing is only half of the inner ear's responsibility. Notice these 3 looped structures sitting above the cochlea, the semicircular canals. Each ear contains 3 of these canals, placed at exact right angles to one another, mapping directly to 3-dimensional physical space. When you move your head, physical inertia displaces the endolymph fluid trapped inside these canals. As the fluid moves, it stimulates nerve endings housed in enlarged ends called ampullae. This generates the precise spatial sensation required for the brain to maintain upright posture and equilibrium. Because these transduction engines rely on exact physical tolerances, microscopic structural failures inevitably result in functional loss. If the aqueous humor in the anterior chamber of the eye fails to drain through the canal of Schlemm, internal fluid pressure builds. That pressure pushes backward, slowly crushing the optic nerve in a condition known as glaucoma. As the crystalline lens loses its shape, it cannot focus images precisely onto the retina, generating refractive errors like myopia or hypermetropia. In the inner ear, disruptions to the delicate fluid dynamics within the labyrinth cause Meniere's syndrome, resulting in a severe, simultaneous loss of both hearing and physical balance. Looking at these dual pathways, the mechanical precision required to make contact with the outside world is clear. Human interaction with the environment relies on these biological machines successfully translating the universe into neural code. Mastery of human anatomy requires looking past a static list of terms. It demands an understanding of the continuous sequence of physical, chemical, and neural events that allow us to perceive reality.